Hello, good afternoon. Welcome to this ninth session. And for me, it's a real pleasure to introduce the first speaker, Professor George Perry from the University of Texas at San Antonio. He is Dean of the College of Sciences. Professor of Biology at the University of Texas, and he is recognized in the field of Alzheimer's disease research, particularly for his work on oxidative stress. And today he is talking about in search of a cure for Alzheimer's disease. Thank you so much.
Thank you very much, Eva.
Thank you.
Muchas gracias por todo, por invitación y todos aquí. Yo muy feliz para segunda vez en Salamanca. Anyway, I'll present in English because I will do much better in English. And I'll listen to my wife who tells me not to speak Spanish. Any case, what I'm going to focus on is really work on oxidative stress and its source and how it provides some understanding of why it may be very difficult to have an effective therapeutic for Alzheimer's disease. And maybe some ideas I speculate on why we've gone wrong so long just by reversing concepts about whether proteins are causing the disease or protective responses to the disease, which violates the usual rule of dividing the plaques and tangles as the cause or irrelevant. I'm going to take something which nobody has, that they're really important, but they may not be the cause. Okay. So the work I'm going to present is a collaboration of many different people. You know, from the very beginning, my late friend and colleague Mark Smith and Larry Sayre, who helped with a lot of the chemistry, and continuing along was Jean-Wei Zhu and Xinglong Wang at Case Western, and at UTSA, Andrea Kelly and others. And the work I'll present toward the end about metal redox speciation with Joanna Collingwood at Warwick University using facilities at Oxford. And in various studies, I've worked with many others, including Eva, over the years. Okay, so where do we start in Alzheimer's disease? You could say we started in 1906 with the index case, August Eder, and plaques and tangles. That was the definition: dementing illness, loss of cognition, and amyloid deposits and tau deposits. But why haven't, if it was so simple as having these abnormal things, in other words, a normal person doesn't have them, at least when they're young, and you have this pathology associated with the disease, why is it that, from my analysis, is about 60,000 people over the last 20 or 30 years have been involved. And these are not people without talent, including people now in the audience. 116 years of study, tremendous amount of money. The clinical estimations, Tom estimated, Sudhoff, $4 billion, but my estimates are it's more like $40 billion has been spent in clinical trials. The United States government is spending about $4 billion a year right now. And yet, the failure rate for drug development is over 90%. The only drugs that actually are being used widely are symptomatic and are borderline in their effect. And is it because the focus has been too long in plaques and tangles? Or, let's see if I can use this. Will this go up there? Yeah. Okay, that's easier for me. Is it because too long and we've been focusing on removing the plaques and tangles, and that'll the idea I'm going to develop. And also, with the most recent studies, whether Aduhelm or the more recent study that was just mentioned within the last week, studies of autosomal dominant Alzheimer's disease among people in Colombia, that there was no clinical benefit of removing the amyloid. And along that line, saying it was too early or too late or whatever it is, I have an answer to that, what I can tell you later, is treat in utero, and you might do better. Obviously, you're never going to be able to treat earlier than 10 years before the disease in any practical way. So where do we go there? Do we move away from amyloid? Or we go to tau? Do we have other concepts? Okay, so right now there are a number of targets. This is from a review we took, but I've had several reviews I've written about reviewing clinical trials in the last few years. And none of them have had really major success. This probability of success is some other people are doing. So most of the clinical trials right now still are working. The ones that the large phase 3 trials tend to be amyloid related, some are tau related, and then there's other types, inflammatories, etc. So far, none of these have really shown to be truly effective. So now I'm going to introduce the concept of oxidative stress. And Luke mentioned to me something, you're a marine biologist. Well, that's where I started. 47 years ago, I started my career as a marine biologist, and I studied oxidative stress at fertilization, the beginning of life. At the time I began these studies, I was looking at the classic definition of oxidative stress, and that's the one that is simplistic. Oxidative stress is breaching antioxidant defenses. It's part of us breathing air. For the last billion years, or near a billion years, there's been tremendous oxygen in the environment that leads to us metabolizing and burning our food completely to carbon dioxide, but that oxygen also reacts with us, and our body has tremendous defenses against it. So you could call this the model of the rusty nail. When you're born, you're reduced, and as time goes on, as you get older, it begins more and more rusted. Well, the evolving definition, I think I learned from the time I was a student at fertilization. If sea urchins do not have an oxidative burst, they don't develop. So the evolving definition is that actually things are always in balance. What changes is the different homeostatic balance. In other words, there's antioxidant defenses. I won't talk about all of them, but there are many of them. I will focus on tau and amyloid as them, but there is also glutathione and many other things that are involved. And that's why it's not as simple as just taking vitamin E and vitamin C. Okay, so what was the evidence for oxidative stress in Alzheimer's disease or oxidative damage? So this is one of the latter of the studies we did, and it's looking at, are there modifications of molecules related to oxygen in Alzheimer's disease? And we used an antibody to guanosine 8-hydroxyguanosine, which recognizes both DNA and RNA oxidation. And you can see that a section through the hippocampus in Alzheimer's and control, there's quite a difference. And as you see this at higher magnification, you see the cytoplasm is really robustly stained, and you could think that's mitochondrial DNA. I will tell you that it's actually mostly ribosomal RNA, and the cytoplasm is stained, and the nucleus is actually consistent with the ribosomes. The nucleolus is stained. Now this is a neuron with a neurofibrillary tangle. It's black and white, but it's birefringent here. And this neuron with a neurofibrillary tangle, the remaining cytoplasm is less intensely stained, actually by half. So neurons that have tau pathology actually have reduced oxidative changes. Well, that staining pattern, and why I cut to it, is because initially we really focused on the lesions, thinking that plaques and tangles, they have long residence time in the brain, so they'd have lots of chances to get modified. When we started to look at incident changes, such as nucleic acid oxidation or protein oxidation, we found that the dominant place the oxidative damage was in the cytosol of vulnerable neurons, the neurons that are likely to die. And I draw here a timeline. So I have a normal neuron changing eventually to a neuron that has a tangle, and prior to then, the accumulations of tau, and eventually that neuron dying and being an extracellular neurofibrillary tangle. Tau would be present in this type of neuron, where there's pre-tangles, and it would also be present after the neuron dies. The oxidative stress or damage occurs in neurons that have no tau abnormalities. And I put a question here. This neuron is a neuron that's in a person with Alzheimer's disease but looks normal. And this was quite a paradox for us because there really wasn't any described abnormality in these neurons. And therefore we looked at what could be possible in generating it. And we looked at mitochondria, and we looked at amyloid beta, and we looked at redox active metal changes. And why we looked at that, is because the broad type of oxidative damage, I guess I should go back to this. This is changes in sugars, which are sugars react with proteins. And I might add parenthetically that that circle of tau that Michelle showed earlier, we showed earlier that bringing together oxidative modifications probably plays an important role in it, in cellular cross-linking, either with sugars or with other carbonyl type changes. This is protein direct oxidation, and this is lipid peroxidation that react with reactive groups. Okay, and then, so to have this broad type oxidation restricted to the cytosol means that you probably have Fenton type chemistry occurring. That is the catalysis of iron or copper causing redox cycling. This type of cycling, and I said type, is actually really important in all of our bodies. It's important in metabolism. The electron transport chain is basically changes in iron and copper redox cycling and lowering the energy. In this particular case, you're transferring the electrons and removing them from other molecules and oxidatively modifying them. And if you're creating something like this, an hydroxyl radical, it will react with the first adjacent molecule it sees. Okay, but to know if this is really happening, you have to be able to establish all these redox types of metals. And I'll show more about this, that we've now been able to do this directly. So one of the ways we did this initially was looking for redox active iron accumulation in cells to know if there was a change in it. And what we did here was look into a redox reaction. And this is a case of Alzheimer's disease, and this is a control case. And we found in neurons and in plaques that there was the ability to change the redox properties of iron and copper and reduce and oxidize, either reduce or oxidize iron. We actually showed, and I won't show the slide here, that this is a really early change. It occurs during MCI, that the neurons have this type of change. And this led to the following idea, underlying hypothesis, that actually during Alzheimer's disease, there's a change in copper and iron redox activity, not an increase in copper and iron. They are not there's no indication that they really are majorly increased. What changes is how they're organized. And we thought that they were likely to coming from the turnover of mitochondria, dealing with autophagosomes and lipofuscin playing a really critical role in this. And I won't present all of the data we have with lipofuscin, which we think really is an important part of this picture. So mitochondria themselves release oxidants, even as part of their normal function. They release superoxide. Just like cars are not, well, gas-powered cars, electric cars are another circumstance, but gas-powered cars are not 100% efficient. We are not 100% efficient with the oxygen we breathe, and some of it goes into forming superoxide, which is usually detoxified. But if, before being detoxified, it interacts with redox active iron and copper, it'll oxidize other molecules. This requires, again, that we identify what type of mitochondrial abnormalities there are and metal redox changes. So to look at the mitochondrial changes, we did in situ hybridization using mitochondrial DNA. Initially, we were looking for mitochondrial DNA abnormalities, and we did in situ hybridization. We found those abnormalities, but the most striking thing we found was an increase in the amount of mitochondrial DNA. And you can see that here, that this is an aged person, and this is somebody with Alzheimer's disease, and it's about a 3 to 4-fold increase in mitochondrial DNA. If we look at mitochondrial proteins, we just see the same increase, although it is different in localization. It's more diffuse within the cytoplasm. But both protein and DNA are increased, and this led us to do a study of biopsy tissue to understand, are there really more mitochondria in Alzheimer's disease, or are there more something else? When we did that, we found, if anything, there's a reduction in normal-looking mitochondria with double membranes, that there's no real major change with mitochondria with cristae. That's in the publications. People talking about broken cristae. These are fixation artifacts, and tissue fixed to biopsy is not immersion fixed. I mean, it's not perfusion fixed, so you end up with artifacts. And there was an increase in baculation associated with lipofuscin. Okay, and why do we bother to look at lipofuscin? Because we noticed when we looked, where is this mitochondrial DNA? Well, of course it's in mitochondria, but the dominant place we saw mitochondrial in situ hybridization was in autophagosomes. You can see here with EM, with gold nanoparticles attached to the in situ probe, that it's really quite apparent that it's mostly in these particles, but it's also present in the cytoplasm, and I'm not showing here, cytochrome oxidase is almost all in the cytoplasm. So there's a whole bunch of garbage derived from mitochondria in neurons. And this changes during the aging process and really is pronounced during Alzheimer's disease. And highly relevant to what Ken Kosek presented earlier, means that there's a lot of lipids in membrane fragments in the system, may be important in initiating tangles. Okay, so then we looked at the issue of mitochondrial dynamics, trying to understand why is all this mitochondrial junk here. Together with Jesus Avila, we looked at degradation, but I won't present that. There's alterations in how mitochondria are degraded. And ourselves, we looked at both the issue of microtubules together with Jesus, and also fusion-fission, because mitochondria are extremely dynamic and quite important for a neuron that has long axons and dendrites to be able to move them, because the proteins are mostly made near the nucleus. So they move out anterogradally away from the cell body, down axons, and they return back retrograde, or they fuse. So a neuron can have 1 mitochondria or 1,000, it still could be the same amount of material. So when we looked at microtubules to understand if that was really a major feature, we did this looking at the biopsy samples to look whether there were changes in microtubules. And we found something we think is pretty surprising, that if you looked at neurons from control individuals, they have much higher microtubule density than those people with Alzheimer's disease. And further, there's no difference whether there's a tangle there or not. 0 difference. We have an internal control for this because we have smaller neurons that are less affected in the disease, and there's no statistical difference. And you can see here it's a huge statistical difference, even with the small samples. And this is just showing an electron micrograph. And when you can see a neurofibrillary tangle filled with paired helical filaments and microtubules in this same micrograph, it's not a rare event either. So people with Alzheimer's disease probably have an 80 or 90%. This probably marks a lot of this during the aging process, because even with a small number of cases examined, we found a decrease in microtubule density from aging. We weren't going to even show this graph, but I showed the statistics, and the reviewer said if you had an R squared of 0.98, you had to show it, even if it's N of 4. Okay, so there's proteins that regulate mitochondria. Oh, so I might add, we did that in an experiment. We actually tried modeling that in cells, inhibiting microtubules. We did not see the same mitochondrial changes by inhibiting microtubules, as opposed to looking at fusion-fission, which appears to be more rate-limiting. And just as microtubule assembly and transport is well regulated, so is fusion and fission. I'm going to focus on one protein. We've examined essentially all of them and their role. But I'm going to focus on this one dynamin-related protein or dynamin-like protein, which is important in breaking mitochondria apart. If you look at this in, let me see if I have, in it's common in the same neurons that have the oxidative stress, and if you look at protein levels, you'll see it's reduced by about 75%. Okay. And what was interesting, we were able to find a same pattern if we looked at fibroblasts. These are fibroblasts in culture derived from Alzheimer's patients and controls, and we found about a 50% reduction. We also found if we culture these cells that they have this same issue of collapsed mitochondria. It's not all the cells, but it's more common in, you can label, these are living cells, so now you can label them with vital dyes for mitochondria or dynamin-like 1 protein, and in Alzheimer's cases, about 20% or so have this collapsed morphology. Among normals, it's a very low number. Okay. And you can change the morphology just by changing dynamin-like 1 protein. If you reduce dynamin-like 1 protein with either inhibitory RNA or dominant negative, you end up with the same collapsed morphology. And you can rescue them also by overexpressing. So it appears to be an important element. Now, what changes this pattern? Well, one possibility, well, certainly it plays a role. I can't say it's the only one. Oxidative stress plays some role in it, but amyloid production plays an important role in this dynamics. Because if you take the cells and you overexpress wild-type amyloid protein, you end up with more of the collapsed mitochondria. This is in normal cells. And if you have the Swedish mutation, you even get more. You're getting over half the cells. And if you block production of amyloid, you block it. And those cells overexpressing APP, you can actually do a test to understand what effect it has on mitochondrial dynamics. If you bleach a region of the cell and you watch it recover, if you're expressing the APP, it actually blocks that about threefold. So this part of my talk actually suggests amyloid plays a really critical and important role in mitochondria and may even have a causative role for what's happening here. So the normal situation in a normal individual is the cell, the mitochondria are distributed throughout the cell. And in somebody with Alzheimer's disease, instead, you end up with this paranuclear clustering of mitochondrial garbage. And if you look in axons, which we did in biopsy sample, the proximal region of axons, and we found that there was a lower density of mitochondria there. But what do you, when you examine this further, then what do you actually see for the 8-hydroxyguanosine and amyloid relation? So I'm going to show several things here. First, Down syndrome, sporadic Alzheimer's disease, and hereditary Alzheimer's disease. And they all have similar patterns. I think each is informative to the other. So if you look at Down syndrome, the blue stain is 8-hydroxyguanosine. It's marking basically oxidized ribosomes, and they're oxidized due to bound iron. We showed that in other studies. And so these neurons are stained in the cytoplasm, and this is a teenager with Down syndrome. And why we did Down syndrome, not some the aging changes are much more predictable. So somebody middle-aged with Down syndrome, you see plaques, in the teenage very little plaques, and you see it's much fainter. And somebody who's 60, you can't even see any staining in the neurons. Again, kind of surprising. So as Down syndrome progresses, actually the oxidative damage is reduced. And this is, all of this quantitation is on a per neuron basis, done by specific densitometry. So if you quantitate this, and break it up into quartiles, you'll see the oxidative stress, which is in the bar, goes up way earlier than the amyloid, like at least a decade earlier. So it predates amyloid. And notice that the oxidative stress is going down when the amyloid's going up. So those cases with amyloid, we do the correlation, and it's an inverse correlation. So the same thing happens, so amyloid, more amyloid, less oxidative stress in the remaining neurons. And here we're looking at what I showed you before, but the quantitation of it, 8-hydroxyguanosine in neurons with tangles versus not. And you see it's reduced by about 50%. And in that case, we have a direct explanation, or at least one explanation. Wherever tau is formed within neurons, in every case we've examined, heme oxygenase is present. Heme oxygenase is an enzyme that degrades heme, one of the prosthetic groups within mitochondria, and changes it into an antioxidant and free iron. So this is heme oxygenase. Let's see, well, anyway, heme oxygenase here, and those are those little black dots. And this is a hippocampus of a normal person. And you see there's none. And if it did it, it totally overlaps tau staining. And in fact, we showed in cell culture models, if you overexpress heme oxygenase, it changes tau phosphorylation. Okay. So further, okay, so that explains it for tau. Just the induction of that enzyme would be critical in changing oxidative balance, and because it's a well-known antioxidant. And then when does the oxidative damage occur during Alzheimer's disease? Sporadic form, and we collaborated with Washington University for their incident cases. And you can see here that the increased, number one, it's specific for large temporal parameter neurons, and it's highest during MCI. So it's an early change. So this is more typical case of a broader range that we analyzed in Cleveland of cases of Alzheimer's disease and looking at amyloid burden versus oxidative damage, and you see the same relationship. More amyloid, less oxidative damage. It fits a time curve because the cases, in other words, people were diagnosed and died sometime later. If they lived a longer time period, they tended to have more amyloid, but they also tended to have less oxidative damage. Oxidative damage in sporadic Alzheimer's cases of long duration goes back to normal. Okay, and the p-values are very good, 0.002. There's a slight difference with ApoE because people with E4 have more amyloid deposits with time. In case of familial Alzheimer's disease, we see the same relationship. That it's an inverse relationship. I'm showing it here for amyloid beta 1 to 42, and that we did that for all the other studies. Also, in the inverse relationship is always amyloid that ends at 42, and not for that ends at 40. We also analyzed here an asymptomatic case that died of a heart attack before the age at risk. They had limited pathology, but very high oxidative stress. There's no error bar because it's an N of 1. So in all the cases of Alzheimer's disease, all the cases we've examined, and I can talk about the one minor exception, which probably is important. Well, I shouldn't say minor, one exception. It happens. We've looked at what would be the mechanism for amyloid reducing oxidative damage. Well, we've looked at isolated plaques that can be very easily isolated. And this is with Congo red stain, this is with an antibody stain. And we did look for metal binding using Raman spectroscopy. And we found that they had a copper bound to them and zinc bound to them. Those are those peaks there, those little peaks. But we couldn't find any sign of iron, although iron is present. And why were we interested in copper? Although think one thinks of copper as a really inert metal, copper actually redox cycles much more actively than iron, and cells usually control copper much more than iron. If you look at the ability of a fat or ascorbic acid, vitamin C, to be oxidized, which both of them will, in water, they do so very slowly. Copper will catalyze their oxidation just by itself. And that's what's shown here. In this case, it's an oxidation of arachidonic acid to form hydroxynonenal, which is a product of lipid peroxidation. And if you had just pure copper, that's what you'll end out with very low levels. If you add 1-to-1 amyloid beta, you essentially block it. The amyloid binds the copper very strongly, and you don't see any redox cycling. So this led us to really want to understand how the copper is organized and how the iron is organized within plaques. Not an easy task. We're still on that path. So this is one of those studies. And it was done in atomic resolution electron microscopes, spherical aberration, so you can see individual atoms. The other part that's critical is that these specimens, these are plaques that are embedded in tissue. The tissue is not treated with any heavy metals, so there's no lead or uranium or osmium in this sample, and the electron contrast is really produced by the endogenous metals predominantly. And you can see these little dots that are marked with yellow here that actually, even in standard methods, you might notice. Okay, when we do look at those very carefully, we see that they have spectra, and you can actually see individual atoms. That's actually what's shown here, individual iron atoms, and those structures are exactly those of magnetite. And then in a more recent, they have magnetite, and further, I'm not showing here, we used two different methods to show they had super paramagnetic properties. We also used a X-ray microscope. X-ray microscopes are hooked to synchrotrons, the one at Oxford University or at Berkeley, and then we're able to directly find the metals and directly know their redox position. And we were able to identify what I really wanted to know was whether there was copper 1 and copper 2 pairs, and iron 3 and iron 2 pairs, which really we know already from the magnetite, because that's what it is. But we found, really surprisingly, is that we found a lot of metallic metals. In fact, they were the dominant forms. Iron 0 and copper 0. This is the first time they've been identified in the human body to our knowledge. In fact, in any animal body. Okay, so we found all the components necessary for redox cycling, and surprisingly, we found metallic metals. Okay, this is some more things. Anyway, all of these are just analysis. Oh, I do have this is another magnetic circular dichroisms. So we've used several methods to show that they have magnetic properties. And if you take regular plaques, and this amyloid plaque, and this is SEM type approach, and you treat it with a copper chelator, it will fall apart. Although it doesn't appear to fall apart to individual monomers, it appears to fall apart into protofilaments. But we're still working on this. Okay, and then what could be responsible for the formation of, I will finish, I only have a few more slides. Actually, I think 2 more. Is that the amyloid beta actually can undergo redox cycling itself and will change the redox state of copper, and that's shown by a mass spectrometry approach. And so we've identified all the components of Finton reaction present. And this fits along with something Mark Smith and I proposed over a couple decades ago, that amyloid beta and tau, instead of the passive in the disease, or the main drivers, are really critical protective responses to neurons, particularly focused on oxidative stress. So I presented a lot of work related to metals, oxidative stress, et cetera. But I could have presented a lot of other abnormalities, and so is everyone else here. But is correcting any one of those abnormalities going to cure the disease? And in fact, it's led to another idea, is that Alzheimer's disease is a homeostatic disease, one in which the abnormalities mark a new homeostatic balance, which is still viable in the sense the neuron lives. Could it be similar to hibernation? And targeting any of the abnormalities actually is not the right approach because it imbalances the responses that are there. Okay, so you can read the summary slide yourself because I covered it all, right? So I'll be on time. Yeah. Thank you.
Thank you so much. We don't have enough time, but maybe you can ask in the coffee break. Thank you so much, George. Now,